Think we're ready to get started. Thanks everybody for coming. It's my pleasure to introduce Dr. David Sabatino, who's our speaker today, coming from Montreal, Quebec, Canada, where he completed his Bachelor of Science in 2002 and PhD in 2007 from McGill. The PhD was focused in nucleic acid chemistry and biochemistry under the supervision of Professor Masad Dama, who's well known in this space as a Canadian nucleic acid scientist. As a research fellow, he completed his postdoctoral training with Professor William D. Lubel at the University of Montreal. I'm not going to try and say that in French, but he's actually a very well-renowned chemist and synthetic chemist, peptide chemist at Montreal, where he worked in developing new synthetic methods to design peptide mimetics. Okay. He was then appointed faculty in the Department of Chemistry and Biochemistry at Seton Hall University, big athletic school in the US in Jersey. He'll a joint appointment in the Department of Medical Sciences at the School of Medicine, where he engaged in biomedical research collaborations. In January 2022, Dr. Sabatino joined the faculty at Carleton University in Ottawa, where he's currently associate professor in the Department of Chemistry and the Institute of Biochemistry. Okay, so he's actually mentored more than 60 students already in his research lab between the two universities, 35 undergraduates, 12 PhD students already, taking them to those terminal degrees. His research interests span the area of nucleic acid and peptide chemical biology. He actually just published a really nice paper in Chem Biochem on synthetic natural killer cell engagers for tumor immunotherapy using multicyclic peptides. Okay, so that's something that's of huge interest in the CDCR and the Immunology Research Center. He's a co-author of more than 40 peer-reviewed scientific publications, conference patents, and a book chapter as well. So, without further ado, it's my pleasure to introduce Dr. Sabatino.
Thank you. Thank you very much. Thank you. All right. Thank you very much, Anthony, for the very kind introduction, for the invitation to be here today. And I also want to thank all of the faculty and students that I had the honor of meeting with today. There were very pleasant discussions that we have, very exciting research that's going on here. And it's just a pleasure to have the opportunity to be here today and to tell you a little bit more about our research program geared towards the title of today's presentation, targeting the glucose-regulated family of chaperone proteins for applications geared towards cancer therapy. So today's presentation will be divided into two main research themes, both of which are correlated towards targeting the glucose-regulated proteins for anti-cancer applications. I'll begin by highlighting to you our applications in RNA chemical biology as synthetic tools, potentially geared towards cancer gene therapy applications. And I'll highlight representative examples related to that research theme before transitioning into our second main research area, one based on our work on peptide chemical biology, once again targeting the GRPs for applications based on cancer gene therapy and immunotherapy, before presenting some closing remarks and opening up the floor to a discussion. So let's begin. Our biological targets are related to the glucose-regulated family of chaperone proteins that are ubiquitously expressed in all cell types and primarily function as endoplasmic reticulum chaperone proteins, assisting in protein folding events, especially during cellular conditions of physiological or pathologically induced stress. In fact, in cancer, the GRPs are overexpressed, and in selected cases, they're also presented at the cell surface, where they function as signaling hubs, triggering cancer cell proliferation, adhesion spread, metastatic activity, and even resistance towards drug treatment conditions. Therefore, the GRPs have been classified as clinically relevant biological markers for the development of cancer-targeted diagnostic and therapeutic intervention strategies. Our therapeutic intervention strategy, based on cancer gene therapy applications, is based on the RNA interference pathway, for suppressing malignant oncogene expression. In this gene therapy pathway, synthetic short interfering double-stranded RNA is combined with a transfection reagent, typically a cationic lipid, a cationic amphiphile, enabling nucleic acid condensation and penetration across cellular membranes, where siRNA is then released within the cytosol of the cell, recruited by a protein complex referred to as RISC. RISC not only has binding capabilities to siRNAs, but it also has selective ribonuclease activity, capable of degrading one of the two strands, leaving behind a single-strand antisense RNA, which contains Watson-Crick base pairing fidelity to recognize and hybridize to a complementary messenger RNA sequence. In our case, we're targeting the GRP messenger RNA oncogenes, thereby inhibiting protein translation. Furthermore, as mentioned, RISC does have ribonuclease activity, so it can degrade the messenger RNA strand, essentially eliminating any possibility of the oncogene to be expressed into oncoproteins in the hope of stimulating anti-cancer activities. Despite their potential advantages in cancer gene therapy applications, there are a series of limitations that prevents synthetic siRNAs from being translated into clinical cancer gene therapy applications. So part of our original research program was geared towards improving the gene therapy activity of short interfering RNA. And we rationalized that the incorporation of a branch point molecule, a synthetic anchor that can covalently link both complementary strands, would confer a much more stable, hopefully long-lasting synthetic siRNA construct. We also rationalized that multiple incorporations of these branch point units would allow us to branch multiple siRNA configurations, either targeting the same or multiple oncogenes of interests, in the hope of enhancing the oncogene knockdown effect and stimulating a more pronounced anti-cancer response. So we initially developed a solid phase branch and hyperbranch RNA synthesis strategy based on the automated solid phase synthesis of a synthetic RNA strand tethered to a control pore glass solid support. We then incorporated a synthetically generated orthogonally protected branch point phosphoramidite that functioned as our branch point synthon that we can selectively deprotect and extend our linear RNA segment into a V-shaped construct, and then selectively deprotect the 5' handle of our branch point synthon and continue chain extension to create our Y-shaped branch synthetic RNA. And we can continue and repeat this synthetic strategy for the divergent growth of branch and hyperbranch RNAs, incorporating multiple branch point positions to grow multiple siRNAs, all embedded within the same molecular structure, in the hope of once again potentiating oncogene knockdown responses that can trigger more potent anti-cancer utility. So we tested the applications of the branch and hyperbranched siRNAs in cancer gene therapy by preparing a formulation with lipofectamine, a commonly used, commercially available transfection reagent that we use for siRNA delivery within the HepG2 hepatoblastoma, a pediatric form of liver cancer that also exhibits GRP78 overexpression and cell surface presentation, that we rationalized upon siRNA delivery can be used to shut down GRP78 expression, that we can then probe with incubating the cells with a fluorochrome labeled anti-GRP78 antibody, and visualize the dimming of the green fluorescent signaling as a function of reduced GRP78 presentation. We also confirmed whole cell levels of GRP78 knockdown by Western blot, in which the more potent branch and hyperbranched sequences that contained multiple sRNAs that can target multiple GRP78 messenger RNA sequences resulted in the most potent knockdown responses, generating 40 to 60% GRP78 knockdown, more significant when compared to the linear controls, even when added in combination. Unfortunately, GRP78 knockdown only resulted in about 15 to 20% cell death, underscoring the resiliency that tumor cells have, especially towards GRP78 knockdown. So in a sense, we went back to the drawing board and asked ourselves one very pivotal question. What can we do to improve the anti-cancer utility of our high-ordered sRNA structures? And we came up with two ideas. One was based on the assembly of sRNA nanostructures, potentially using the branching RNA modality as a template for the self-assembly of complementary strands to generate these self-assembled sRNA nanostructures that now incorporate multiple sRNA motifs, all embedded within the same molecular structure to potentially knock down multiple GRPs in a combination approach that we hoped would not only improve GRP knockdown efficiency, but also anti-cancer responses. We rationalized the same effect through the application of sRNA bioconjugates, in this case, combining the cancer gene therapy effect of the sRNAs with the anti-cancer utility of some payload in order to improve the anti-cancer responses, once again, in a combination therapeutic strategy. So let me highlight one representative example of each class. Here's a representation of the self-assembly of linear V and Y-shaped RNA templates, synthetically generated as previously described, and then using them as templates to combine complementary linear strands to afford the linear V and Y-shaped sRNA hybrids that can combine single or multiple sRNAs within the same molecular scaffold. We also developed a self-assembly strategy in which we combined complementary and stoichiometrically regulated quantities of V and Y-shaped constructs to afford these genetically encoded geometric shapes, resembling a variety of different polygons that can combine multiple sRNAs within the self-assembled nanostructure formulation. Once again, with the goal of improving GRP knockdown efficiency and the hope of potentiating the cancer cell death response. Transmission electron microscopy revealed and confirmed the discrete, monodisperse, self-assembled sRNA nanostructures that mimicked a variety of different polygons, ranging from pentagons, squares, rectangles, hexagons, and porous spheres that were monodispersed in their distribution and of restricted sizes, ranging from about 50 to 150 nanometers in size, making them applicable to molecular cell biology applications. So we screened a library of about 20 sRNA nanostructures against the AN3CA endometrial cancer cell line, which exhibits addiction to GRP overexpression, and found the most potent anti-proliferative responses associated with the self-assembled sRNA nanostructures that can target multiple GRPs in synergy. In fact, one of our leads belonged to this Y-shaped construct that incorporated 3 sRNA modalities, all embedded within this branch molecular scaffold that can target all 3 main GRPs in synergy in the hope of producing a synergistic anti-cancer response. We tested the activity of this construct across the endometrial, breast, and cervical cancer cell line and found the most significant knockdown responses associated with this Y-shaped structure across the panel of tumor cell lines, which also resulted in the most significant anti-proliferative effects across the panel of tumor cell lines, and this time stimulating anywhere from about 50 to 60% cell line toxicity once again across the panel of tumor cell lines tested. Interestingly, we also tested the activity of this construct against a non-tumorigenic MRC5 control cell line, which displayed very little cell line toxicity in blue as a function of reduced GRP knockdown and very little, if any, significant anti-proliferative effects being observed, underscoring an element of treatment specificity that's associated with these constructs, being able to impact tumor cells that display an addiction to GRP overexpression, and to a lesser extent in normal healthy cells that display normal basal levels of GRP expression. Now, moving towards a representative example of our efforts in RNA bioconjugation for generating the so-called siRNA cancer theragnostics, combining both therapeutic and diagnostic utility to our linear V and Y-shaped constructs that can contain either a single fluorescent reporter or multiple fluorescent reporters to enhance cell-based detection, potentially geared towards preventative cancer treatment strategies. So we initiated this project by developing a solid phase synthetic RNA bioconjugation strategy in which fluorescein isothiocyanate was covalently ligated onto alkylamino-linked linear V and Y-shaped RNA templates under mild conditions. We can see decent conversions with linear constructs, but weak, little if any, conversions being observed with the more sterically encumbered V and Y-shaped RNA templates, thus forcing us to develop an alternative self-assembly strategy in which we combine fluorescently labeled linear RNA construct with their complementary linear V and Y-shaped RNA templates to afford the linear fluorochrome labeled siRNA bearing a single fluorochrome reporter and targeting a single GRP, the V-shaped construct targeting two GRPs and bearing two fluorochrome reporters, and lastly, our Y-shaped motif targeting all three main GRPs and incorporating three fluorochrome units within the same molecular scaffold. We then tested for cell-dependent uptake, GRP knockdown, and anti-cancer utility within a PC3 prostate cancer cell line in a time-dependent manner to examine the extent of cell line detection, followed by GRP knockdown and cell line toxicity. And we observed cell-based detection according to flow cytometry. The linear siRNA bearing a fluorochrome reporter exhibited a population of labeled cells over an extended 3-day incubation period, according to flow cytometry, which was also supported under the confocal microscope. Under shorter incubation periods of 4 and 24 hours, we can see some cells being labeled with the fluorescent reporter containing siRNAs. We then compared this control with our V-shaped construct bearing 2 fluorochrome units, and under the same 3-day extended incubation period, found a larger population of labeled cells according to flow cytometry, and under the confocal microscope, a larger detection of fluorescently labeled cells over a 1-day incubation period. And lastly, our Y-shaped structure incorporating 3 fluorochrome units, all within the same molecular structure, exhibited the largest population of labeled cells, according to flow cytometry over that same 3-day incubation period. And under the confocal microscope, cell imaging and detection really lit up over that 24-hour incubation period, underscoring the ability to enhance cell-based detection by including multiple fluorescent reporters within the same molecular motif, potentially giving us molecular probes that can facilitate the early onset detection of cancers in order to establish preventative, more effective therapeutic intervention strategies. And because there's an element of therapeutic utility that's genetically encoded within these RNA bioconjugates, we validated the ability for these RNA bioconjugates to shut down GRP expression within the PC3 prostate cancer cell line, either containing a single or multiple fluorescent reporters. And we also validated and correlated GRP knockdown with cell death events. And the prostate cancer cells are one of a handful of cells that are quite adherent as a mechanism that's associated with prostate cancer cell adhesion, spread, and metastatic activity that makes them much more resilient towards treatment conditions. In fact, we found a significant population of dead cells floating in the supernatant of the cell culture media, suggesting that there may be an impact in reducing cell-based adhesion within the cell line upon GRP knockdown that can make this cell line a little bit more sensitive towards therapeutic intervention strategy and sensitizing them to cell line toxicity. So we were interested by this observation and decided to further explore whether or not there was a molecular interplay, a correlation in between GRP functional biology in tumors to that of the adhesion proteins, and to see whether or not there was a molecular link in between the main GRP chaperone GRP78 and N-cadherin, one of the main adhesion markers that contributes to prostate cancer, as well as a handful of other tumors, adhesion, spread, and metastatic activity, especially to bone tissue, where they're not only resilient towards treatment conditions, but are essentially incurable at the later stages of diseases. So here's our study. GRP78 knockdown was found to reduce N-cadherin expression, at least at the protein levels expression, to a lesser extent at the messenger RNA levels of expressing, suggesting an element of post-transcriptional regulation across the prostate cancer and multiple myeloma cell lines. We were also interested in exploring whether or not GRP78 knockdown can sensitize prostate cancer cells and multiple myeloma towards drug treatment conditions, as originally hypothesized. You can see that drug treatment conditions of multiple myeloma cells boosts GRP78 expression levels to a lesser extent and cadherin, but allowing us to now sensitize the cells towards GRP78 knockdown in the presence of siRNA treatment that also allows for concomitant suppression of N-cadherin, now sensitizing the cells to drug treatment conditions, having a more robust cell line toxicity response occurring as a function of GRP78 knockdown, which also reduces levels of N-cadherin. We are always curious to see whether or not GRP78 knockdown impacted other key adhesion biological markers. We could also see suppression of N and E cadherin, including beta-catenin, and we also probed for EMT markers that are associated with metastatic spread, and interestingly found an overexpression of EMT markers TGF-beta1 and SNEL2 associated with GRP78 knockdown, correlating a link between metastatic spread and GRP78 functionality, which resulted in significant phenotypic changes of the prostate cancer cells upon GRP78 knockdown, transitioning from healthy, viable, elongated shaped cells into unhealthy, rounded, dead cells as a function of GRP78 knockdown, underscoring the interplay that GRP78 knockdown has on cell line toxicity by sensitizing prostate cancer cells towards cell toxicity as a result of reduced adhesion. In this case, we performed this assay in co-culture with bone osteoblast cells. So we reduced their adhesion capability to bone cells, rendering them much more sensitive towards cell line toxicity. So I hope I've been able to highlight to you during this first part of my presentation how we can use synthetic RNAs of a mutual architecture and structure as molecular probes, potentially for therapeutic and diagnostic applications. And we're currently interested in developing a cancer-targeted gene therapy approach that can shut down mutant and evasive biological markers, especially for combating treatment-resistant tumors, in the hope of developing a translational, clinically oriented cancer-targeted gene therapy approach. And let me highlight some of the current strides that we've taken in this research area. And this research area is predicated on the identification of a clinically viable biological marker that allows us to discriminate between tumor tissues and healthy ones in the hope of developing a cancer-targeted intervention strategy. As mentioned, GRP78 is a clinically relevant biological marker overexpressed on tumors, and in certain cases presented at the cell surface, but absent on healthy cells, giving us a biological marker to target tumor tissues discriminately when compared to healthy tissues. So we've identified a tumor biomarker that we can potentially target for the development of cancer-selective intervention strategies. How do we target this biological marker? Well, studies have shown the phage display peptide library selection of a small set of GRP78 targeting peptides that also function as tumor homing peptides, primarily having good affinity and selective binding to GRP78, our biomarker of interest. As you can see from molecular modeling and docking studies, there's a peptide binding domain that's incorporated between this helix bundle and beta sandwich subdomain of GRP78. And you can kind of see this binding pocket that facilitates the incorporation of a GRP78 targeting peptide sequence. That's stabilized primarily through a series of hydrogen bonding and hydrophobic interactions. So they're non-covalent bonding interactions that are participating in between this ligand receptor binding interaction. GRP78 targeting peptides have been made, and they've been also conjugated with fluorescent reporters to be able to discriminate between GRP78 presenting tumors and those that lack GRP78 presentation. And they've also been conjugated with toxins and anti-cancer agents, including photosynthesizers that can be useful in photodynamic therapy applications to reduce cell viability selectively in tumors that overexpress and present GRP78 at the cell surface while leaving healthy cells unscathed. Despite their tumor homing capabilities, especially within GRP78 presenting cells, these peptides lack cell penetration or have very little cell uptake capabilities, which limits their therapeutic utility. And we were interested in exploring the ability to improve cell uptake efficiency of these GRP78 homing peptides, potentially for gene delivery applications that can be then translated into our cancer-targeted gene therapy approach. So we've designed the GRP78 targeting peptide sequence to also contain a cell-penetrating and intracellular localization sequence in the hope of improving cell uptake efficiency of the GRP78 targeting peptide, while also enabling an element of intracellular localization. So we could potentially deliver cargo at selective compartments within the cell to administer very specific and site-localized intervention. So we've synthesized a library of amphiphilic polycationic GRP78 targeting peptide sequences following an FMOC solid phase peptide synthesis protocol. This is an automated synthetic procedure that follows amino acid couplings and deprotections on solid phase to elongate your peptide sequence of interest, containing either naturally or unnaturally occurring amino acids. So we can make whatever peptide sequence you'd like on solid phase. Following synthesis, cleavage, deprotection, analysis, characterization, and purification by LCMS gives us peptide sample in hand to complete our structure-activity relationship study. So we primarily rely on circular dichroism spectroscopy as an instrumental tool to give us information on peptide protein secondary structure, and found this unusual helical coiled structure motif, combining elements of alpha helix and random coil helical coiled structures that are associated with these peptides and were found to be thermodynamically stable according to thermal denaturation studies. We also imaged the ability for these peptides to self-assemble into higher ordered self-assembled nanostructures that can potentially facilitate drug delivery applications. Interestingly, these amphiphilic polycationic peptides self-assembled into thin filaments, short protofibrils, and this intricate web of nanofibers that could potentially be useful for drug loading and delivery applications, in addition to some spherical nanoparticles that may also lend themselves to the same type of application. So we did a trial study with curcumin, a polyphenolic natural product that has a wide range of therapeutic properties. We resorted to fluorescence spectroscopy to determine the dose-dependent binding of curcumin to our peptide of interest, which self-assembled into nanofibers in the absence of curcumin, and in the presence of curcumin, those nanofibers were abolished, limiting, obviously, their drug delivery applicability. But compared to what literature has to say, in between the binding events occurring with curcumin and other type of fibril proteins and peptides, this result was not entirely surprising. As we could see, curcumin also has the ability to inhibit fibril formation of other fibril-forming peptides and proteins, including amyloid beta, one of the key proteins that's involved in fibril formation in Alzheimer's disease models, potentially lending curcumin in therapeutic applications in combating fibril-forming neurodegenerative diseases. So our synthetic peptides behave very similarly to amyloid beta and can be potentially used as a simple synthetic model for studying how drugs can bind, interact, and in this case, inhibit the assembly of fibril formation. But that wasn't our ultimate goal. Ultimately, we were interested in improving the cell uptake capabilities of these peptides. So we tested these peptides in the DU145 prostate cancer cell line, one known to overexpress and present GRP78 on the cell surface, and found good binding occupancy of all the peptides that were tested, even during short incubation periods, ranging from 1 to 4 hours. In the dose treatment conditions of the peptides that we were using, typically 10 micromolar or below, there's very little cell line toxicity that's observed, making them potentially useful as cell binding agents, capable of binding to target cells without inducing any toxicity. We also did some cell uptake studies. And interestingly, we resorted to confocal microscopy for this. The FITC labeled GRP78 targeting peptide, so that's the one all the way on top, the W1 control alone. Very little green is observed, highlighting the reduced cell uptake capabilities of the GRP78 targeting peptide alone. Okay, so that was reported in the literature and reproduced by our experiment. We also tested the cell uptake capabilities of our cell penetrating and nuclear localization sequence containing peptides. And these do have, you can see green signaling, an element of cell uptake capability that's associated with these cell penetrating peptides, right? Overlapping with cytosolic and even elements of nuclear localization, which makes them perfectly suitable for drug delivery, gene delivery applications. Interestingly, our test peptides that combine the GRP70 homing peptide with the cell penetrating sequence, R9 or TAT-NLS, restored cell uptake in the GRP70 targeting peptide galactose. So underscoring an element of being able to enhance, improve, or install cell uptake efficiency in peptides that are either weakly or lack cell uptake capabilities altogether. And in fact, we do see an element of cytosolic and nuclear localization of these peptides. We then tested their ability to bind and internalize within cells in a GRP78 dependent manner. After all, these peptides were designed to incorporate a GRP78 homing sequence that gives us tumor targeting capabilities. We did two basic experiments to support this. One was a comparative binding assay in which the fluorochrome label peptides were outcompeted with the anti-GRP78 antibody. And we saw reduced binding of our polyarginine-derived GRP78 targeting peptide in competition with the antibody, good binding in the absence. Comparatively, at 10 micromolar concentration, there's no comparative displacement of the antibody in the presence of this non-specific polyarginine sequence, underscoring an element of GRP78 binding specificity that's incorporated with the GRP78 targeting sequence. We also ran the same experiment under the confocal microscope. We see fluorescent imaging in the absence and presence of the GRP78 blocking antibody with their control non-specific polyarginine sequence. We see reduced FITC signaling in the presence of our GRP78 blocking antibody with the test GRP78 targeting polyarginine peptide, suggesting an element of GRP78 binding specificity that's now been fine-tuned and incorporated within the cell penetrating sequence. We also did inhibitors of GRP78 dependent cell uptake, looking at different chemical inhibitors that may inhibit the GRP78 dependent pathway for receptor-mediated endocytosis. Our control non-specific polyarginine alone and with the presence of inhibitors, especially with the lipid raft inhibitors, either maintains or enhances cell uptake or cell binding of the peptide. Comparatively, our test polyarginine-derived GRP78 targeting peptide reduces or, in certain cases, almost abolishes peptide uptake, underscoring once again an element of GRP78 dependent cell uptake that's associated with our GRP78 targeting peptide sequences. And then the last piece of the puzzle is to see whether or not these peptide vectors can be amenable for gene delivery applications. And there's a large investment being made into the development of gene therapeutics and to overcome their primary limitation, effective cell penetration for gene therapy activity. So we're looking at applying these types of vectors for delivery of a wide range of gene therapeutics in the hope of applying a translational, clinically relevant, cancer-targeted gene therapy approach. And these are preliminary studies that we've accomplished over the course of the last couple of weeks. Our formulation strategy, combining both the peptide with the genetic component. We've done a little bit of optimization work here with plasmid vectors and short interfering RNA, and found that calcium chloride tends to condense nanoparticle formulation, resulting in more condensed nanoparticles as opposed to more amorphous aggregates in the absence of calcium chloride condensation, in the hope of improving cell uptake of the plasmid vector. So we're just doing some optimization studies at this point to see what works best with a reporter GFP expressing plasmid vector. And we're getting some GFP expression with our peptide, but we're still lagging behind the benchmark lipofectamine-based transfection agent. So we're currently in development to try to fine-tune and optimize our peptide-based transfection protocol for plasmid DNA expression and invoking anti-cancer utility in plasmid vectors that are a little bit more related to anti-cancer treatment strategies, and as well for siRNA intervention. So here is peptide delivery of siRNA that results in suppression of GRP78 according to flow cytometry. In the presence of a GRP78 blocker, very little GRP78 expression is knocked down, and it's comparable to the control cell lines that have not exhibited any transfection. We've also confirmed GRP78 knockdown by Western blot, and then cell viability, which, if we're targeting a single GRP, has very little, if any, effect. So we'd like to apply these conditions, obviously, for the delivery of our multifunctional siRNA, our branched or hyperbranched nanostructures that also contain the bioconjugates that we feel may produce a much more potent knockdown response, a much more significant cell line toxicity response in a cancer-targeted GRP78 delivery strategy. All right, and in the last few minutes that I have left, let me present to you one final example of our applications in peptide chemical biology, this time geared towards cancer immunotherapy applications, primarily dedicated towards mimicking the targeting and effector functions of antibodies. On one end, applying peptides that can bind to a certain activating receptor presented on the surface of lymphocytic cells to stimulate an immune response through cytokine secretion. And on the other end, to incorporate peptides that can home in this immune response onto tumor cells that present a tumor-associated antigen, a specific biomarker that will allow us to discriminate tumors from healthy cells. And you already know one half of this equation because I've already presented GRP78 as a biologically relevant biomarker on the surface of tumors, allowing us to home in through the application of GRP78 targeting peptides, a therapeutic response selectively within GRP78 presenting tumors. The second half of the equation is to answer these questions. What is a clinically relevant lymphocytic cell that has an activating receptor that can be activated in the presence of immunostimulatory peptides that can stimulate an immune response and then be directed towards our targeted tumor cell line of interest in the hope of developing the so-called synthetic antibody mimics to mimic the targeting effector functions of antibody through the application of short, simple, bifunctional peptides. So we resorted to the literature in the hope of finding some inspiration on the identification of immune cells and immune targets that we can target for stimulating an immune response through simple synthetic peptides. And we found an interesting study which showed the specific binding interactions, according to flow cytometry, of an Fc NKp30 fusion protein. NKp30 is an activating receptor, activating natural cytotoxicity receptor for the natural killer cells, where upon activation, NK cell killing occurs, especially of targeted tumors. In this case, NKp30 was found to bind with affinity and specificity to a tumor antigen presented on the surface of the K562 leukemia cell line. Chemical cross-linking tryptic digestion proteomic analysis revealed the identity of this tumor-associated antigen, B7H6, as the native protein ligand for binding and activating NKp30 on the surface of NK cells, stimulating a toxic response, especially towards tumors that present B7H6 on their surface. And most tumors do, and this biomarker is absent on healthy cells, allowing the immune system and NK cells to discriminate between B7H6 presenting tumors and healthy cells that lack B7H6 presentation altogether. Unfortunately, tumors evolve to become quite smart and to evade immune systems that can recognize and eliminate them upon recognition. So tumors have evolved resistance mechanisms to shed, eliminate B7H6 from their surface, or reduce the presentation of B7H6 on the surface of tumors, allowing tumor cells to evade NK cell recognition and immunotoxicity, leading to their elimination. They've done binding studies which confirmed a one-to-one binding stoichiometry in between the native B7H6 ligand and the NKp30 receptor, which was dominated by a series of surface loop domains on B7H6, engaging into this hydrophobic binding domain of NKp30 Zooming into one of those loop domains, the FG loop domain of B786, presents the seemingly turn-like geometry composed of 5 amino acid residues, so a pentapeptide motif that contributes to this turn geometry binding into the binding pocket of NKp30. There's low binding affinity. The KD value is about 1 micromolar for B7H6 to NKp30, but there's high shape complementarity. So there's good binding activity of the ligand that is stabilized by a series of hydrogen bonding and cell bridge interactions. So based on the X-ray co-crystal structure analysis in between the native B7H6 ligand and the NKp30 receptor, we mapped out the binding hotspot residues of B7H6 and designed and synthesized short peptide sequences based on these binding hotspot domains, primarily mimicking those surface loops of B7H6 that were engaged into the NKp30 binding pocket, including the FG loop, that pentapeptide that seemed to mimic this turn-like geometry, that we then designed an FG loop mimic that would stabilize a turn-like geometry with this modified FG loop sequence. We also mapped out primary sequence and secondary structure domains and compared their binding activity profiles to a series of negative control peptides. We did some preliminary molecular modeling and docking studies, which seemed to validate the formation of this turn-like geometry with the modified FG loop pentapeptide that seemed to stabilize this turn motif within the conserved binding pocket of NKp30, engaging within the same binding site residues as the native B7H6 ligand, primarily participating in hydrogen bonding interactions. And we also confirmed this turn-like geometry experimentally, using circular dichrosis spectroscopy as a tool that confirmed peptide turn geometry in solution, setting the stage for cell biology. We relied on flow cytometry as a primary tool for determining the binding interactions in between our fluorochrome-labeled peptides, added to the NK cells in a dose-dependent manner, and found comparable binding occupancy of the peptides at 10 micromolar concentrations to the native B7H6 ligand, 1 micromolar, and similar to what we saw with the fluorochrome-labeled anti-NKp30 antibody. So there's good binding occupancy at the peptides at a concentration of 10 micromolar that's also non-toxic to the NK cells. We then confirmed an element of NKp30 specificity in competition with the anti-NKp30 antibody, which outcompeted either native B7H6 ligand binding and reduced its binding capabilities, or peptides by similarly reducing binding occupancy by being outcompeted with the anti-NKp30 antibody. We then ran some ELISA to confirm immunostimulatory activity, if any, and we saw modest immunostimulatory activities of the peptides towards the secretion of TNF-alpha, especially when compared to the native B7H6 ligand. So there's some room for improvement here. We're currently working on that. But in comparing one of the lead peptides here, peptide 6, to a series of negative control, we saw statistically significant levels of cytokine production. Closer investigation of what this peptide 6 actually was is this TVPLN pentapeptide. So that same peptide motif that mimicked that turn-like geometry within the FG loop and stabilized that beta turn structure contributed to binding and improved activation, at least when compared to the other peptides that did not display the same biologically active turn conformation, underscoring the importance of being able to mimic and stabilize biologically active structures, even within short peptidic sequences. So the last part of the equation was bringing all this together. How can we combine our NK peptide epitopes with our GRP78 homing peptide sequences to develop the so-called cancer-targeting immunostimulatory peptides? as synthetic antibody mimics that can home in the immunostimulatory response of NK cells onto GRP78 expressing tumors. And we rationalize that the Y-shaped motif is what Mother Nature presents in antibodies, and we casually described potentially adopting a Y-shaped branching structure to mimic what Mother Nature does in the targeting and effector functions of antibodies. We rationalize that potentially incorporating multiple NK peptide epitopes within the same molecular structure may function to enhance binding and activation of NKp30 presenting NK cells through multiple epitope peptide display and activation of NK cells. We also rationalize that we can link the NK peptide epitopes to our GRP78 targeting peptide sequence that can home in this immunostimulatory response within GRP78 presenting tumors, but also those that lacked B786 presentation. And the first step of this was designing and developing a synthetic strategy for making the synthetic antibody mimics based on FMOC solid phase peptide synthesis that we use to synthesize the linear dipeptides containing two NK peptide epitope domains that flank the central lysine residue that contain an orthogonal protecting group that we can selectively remove, unmask that reactive amino group, and continue FMOC solid phase peptide synthesis to grow our GRP78 targeting ligand to essentially form these Y-shaped bifunctional trimeric peptide as potential mimics of antibodies. We did some binding experiments with the tumor cell lines first. We looked at the HepG2 cells as a model cell line that presents GRP78 and lacks B786 presentation. And what we've seen was modest binding of an anti-GRP78 antibody and significantly more with our fluorochrome-labeled peptides. We were worried about indiscriminate, non-specific binding of our peptides, which was confirmed in the presence of a GRP78 blocker, which effectively blocked antibody binding, but resulted in very little blocking of the synthetic peptide. So this was a bit of a red flag for us, prompting us to explore another cell line to see whether or not it could exhibit more selective binding activity with our peptides of interest. So we then looked at the A549 adenocarcinoma lung cancer cell line that displays more modest levels of GRP78 presentation and absent of B786. So it's a perfect model for what we want to use. As you can see, there's more modest levels of GRP78 detected on the cell line, but to more comparable extent when comparing our peptide binding to the A549 cells and a reduction in peptide binding to the A549 cells in the presence, pre-incubation with a GRP78 blocker. We then looked at the binding occupancy of the peptide with NK cells, which confirmed NK cell binding, and then diminished binding of the NK cells by being outcompeted with the anti-NKp30 antibody, which restored binding, indicating an element of NKp30 binding specificity that was also associated with these peptides. And Luminex also confirmed their ability to secrete inflammatory cytokines, allowing us to then bring all of this together in a co-culture assay to explore whether or not there was an immunotherapy response based on the peptide activated blue Hoyt-stained NK cells directed towards the GFP expressing A549 cells. And we use confocal microscopy to give us indication of anti-cancer utility that was explored with the A549 GFP expressing cells alone, absent of any treatment conditions. You can see that they're readily proliferating. They're quite happy and quite healthy in their proliferation as a standalone culture. In co-culture with the blue Hoyt-stained NK cells, you see both the GFP expressing A549 cells and the NK cells proliferating, doing their own thing without any significant impacts on cell viability or observable impacts on cell biology. That dynamic changes quite significantly when the NK cells are pre-activated with the native B786 ligand or peptide activity. In both cases, you see an expansion of NK cells and a larger association of NK cells clumping the GFP expressing A549 cells together. And in the case of the peptide activated NK cells, you see a dimming and a spreading of GFP signaling, which gave us an early indicator of cell toxicity. So we confirmed cell line toxicity through Luminex by exploring the secretion of inflammatory cytokines across the co-culture conditions. That also resulted in cell line toxicity. Significant early stages of apoptosis were detected across all co-culture conditions, with more late-stage apoptosis being detected in the peptide-activated NK cell treatment of the A549 cells, prompting us to treat and test in vivo activity. So we developed a xenograft mice model of the A549 tumors by initially growing the A549 cells in tumor-bearing mice. Over a 1-week period of time, once the tumors reached sizable geometry or size, we then injected the mice at the localized tumor site with the NK cells alone or NK cells activated with the peptide. And we then monitored tumor regression as a function of time. And these are our results. After the first week period of tumor growth, injection of the mice with either peptide activated NK cells or NK cells alone showed a regression of tumor growth. So the tumors are actually shrinking from day 7 to 11. So there's a 4 to 5 day period that the tumors actually start to shrink upon treatment conditions over a single injection. The tumors do start to pick up again after that day 11 period, but they're growing at a slower pace, at a slower rate when compared to the untreated control conditions. Underscoring at least the ability for the peptides to sustain a small but consistent decrease in tumor growth rate over a 3-week incubation period, underscoring their potential clinical utility in cancer immunotherapy applications. So I hope I've also been able to highlight to you how we can use peptides and employ them either in gene therapy or immunotherapy applications, the latter of which are currently being explored by developing new and improved multi-epitope peptides that can be used to generate the next generation of synthetic antibody mimics, as well as synthetic peptide vaccines. And I'd like to thank my students, former and current, at Seton Hall and Carleton University for accomplishing all of this exciting work. My past and current institutions, Seton Hall and Carleton, for giving me and my students the opportunity to do the work that we love. I also would like to extend a special thank you to our project collaborators and collaborating institutions, as well as funding agencies that have supported past and current work. And last but not least, I thank all of you for your attention, and I welcome any questions.
Got a quick question about some of the very early slides where you were targeting the FG2 cell with some of the earlier generation RNA constructs. I know you use lipopectomy delivered to FG2 cells. What made you choose lipopectomy over conjugating GalNAc to your constructs? Because GalNAc is very specific to liver cells.
Yeah, so we're looking for a commercially available transfection reagent, one that could serve as a good solid benchmark for being able to establish baseline transfection efficiencies. And we saw that with our lipopectomy-based transfections. And then as I presented some of the work getting into our peptide-based transfections, we want to be able to use our peptides, not GalNAc, as targeting vectors for gene delivery and comparing their transfection efficiencies with the readily commercially available cationic lipids, lipopectomy, and some of the others that are commercially available. Yeah.
Can your B788 evolve with the same resistances that these B7H6 can?
Yeah, that's an excellent question. And the answer to your question is yes. So it depends on the cell line, it depends on the tissue in which we're targeting. So before we do any cell biology testing, we always do a little bit of research and literature search to see what's been done in this cell line, in this tissue model with respect to GRP78 presentation. Does it show overexpression or reduced presentation? Is there cell surface presentation that's well characterized, or is that lacking? And if it is, then before doing any of our intervention strategies, we just test the cells to see what are their presentation and expression levels for GRP78 before bringing the peptides and nucleic acids and everything else into the mix. So yeah, that's a very important point. That's something that we're mindful of. And we base either on literature or some of our preliminary studies before getting into it.
Any other questions? We're running a few minutes over, maybe one more. Just give us all a chance, Kevin, just 1 second.
I think she's got a question there. Yeah, go ahead, please. You're welcome.
Question for the ICR part. So the first question is to see when using that to target in different pathways, especially genes with different expression levels, highly expressed versus intermediate, is any consideration to make sure the different target is being knocked down the same efficiency? And the second part related to say, considering the synthesize you are after for that one, but also the skill required and the cost part, would it be possible to using this multiple ICR complex for a systematic screens for testing different genes, like in maybe a screening for
Yeah. So to answer the second part of your question, the answer is yes. We can use these constructs for screening applications, either for screening well-known oncogenes, markers, or for ones that are perhaps not as well-known, elusive or mutants. And I think one of the advantages of this methodology is that the constructs for screening are synthetically generated. All right, so we can synthesize whatever constructs you like with whatever sequence composition you like to screen for whatever biomarker you're interested in. I think that's one of the advantages of relying on synthetic methodology for making these constructs, because you have the freedom and flexibility to design and make and test and screen whatever you like. The cost aspect is also something that we're mindful of, but typically in my experience, the cost of chemical reagents is less when compared to biologicals and the application of synthetic biologicals. For example, using antibodies for doing a lot of this work. A little 100 microliter vial these days is going to cost you $1,000 at Thermo Fisher. Right? We can get multi-gram quantities of reagents for making gram scale quantities of peptides and milligram scale quantities of oligonucleotides at a fraction of cost. So I think that's an improvement as well over the application of more complex synthetic biologics. What was the other part of your question? Or was there another part of your question?
with different expression.
Yeah, so I think the important part there is keeping track of either control genes or housekeeping genes that don't fluctuate in their expression levels as much, and that could serve as a benchmark for comparison, so that we know whether or not we're getting a knockdown or expression response, because it's normalized to a housekeeping gene or a gene of interest that doesn't fluctuate its expression levels as readily as some of these other targets do.
All right, I guess in the interest of time, I think we'll stop it there. Anyone else wants to ask Dr. Sabatino questions, you can hang on for a couple minutes.
Yep.
Join me in thanking for a great talk and very nice to hear.
Thank you.
Very good.
I'm glad it came across.